Okay, good. So again, I'm Jaime Goyesa. I'm glad to be here and joining this seminar for those of you that just did. And I'll be speaking along with my colleague, Mitch Eden. Adam Durando is participating, but he will be mostly speaking during the Q&A session. The same thing goes for a colleague of ours that we invited. Hopefully he's in the audience, Jan Segata from the U.S. Fish and Wildlife Service Caribbean Field Office to answer some questions regarding this translation from science to action. Today, I'm going to be talking about, or we're going to be talking about this long-term engagement to develop strategies to inform climate adaptation in Puerto Rico. The Science Center, this is a multi-pronged approach. It's not just one thing. Today, it just happens that we're going to be concentrated on a case study, the Eleutherodactylus amphibians. And the reason we focus on this group, among the many ways that the Science Center can assist stakeholders in Puerto Rico, is because they are culturally valuable. They do have a role in energy flow in forest ecosystem, tropical forest ecosystems. And above all, they're a sentinel. They're a good sentinel of environmental change, especially climate change. As a group, however, hopefully this works, they are very vulnerable to climate change. So factors affecting them is usually the global warming and drying is probably the first driver of imperil, putting them in an imperiled condition. And that has to do because a lot of key functions, for instance, gas exchange, reproduction, water balance, and behavior are strongly influenced by temperature and water availability. The other one would be habitat degradation. Not only you have an effective loss of habitat, but sometimes when the habitat remains, but it's altered, the microclimatic conditions are changed and therefore not suitable for the species. And last but not least, among vertebrates, Anuran species, amphibians, have very poor vigility or poor ability to disperse, especially from natal areas. As a result, about 40% of these amphibians worldwide are considered threatened. Let me give you an overview here. You have something like over 2,000 species that are considered threatened in this paper that came out in Nature in 2023. The Caribbean highlights or is prominent here as an area of high or hotspot, if you will, of high number of threatened species. Part of the problem is that all of these species, the majority of them anyway, are data deficient. So corrective actions are sometimes hard to get at because we have no idea on how to inform decisions. This group, the Eleutherodactylus, is the group of interest here today, and it's a group from the neotropics, the tropical region in the new world. I'll call it the neotropics, and we have about 200 species that have been described. The majority occurs in the Caribbean, about 84%. A unique feature of this group is that they have direct development. So, unlike the species that you see here in the upper right-hand corner, that need ephemeral or constant water presence in some bodies or some ponds nearby. Uh, these guys will lay their eggs encased in a gel, and this gel provides the moisture, the water that the developing embryos need for until they hatch. So that's very unique. The other thing that had a lot to do with our decision on which groups to use, or what we call representative species, and this is unique from an evolutionary point of view, is that this group of species, this genus, so it's an example of replicate radiation with recognizable ecomorphs. And let me spare a little bit of your time. This is important to us because what it says is that it's a species that develop distinct morphological and behavioral traits as adaptation, right, to ecological niches or habitat, often regardless of their genetic relationship. So when we're asked about when you do these representative groups, are you looking at phylogenetics, phylogeny, if you will? We're not necessarily doing that. We're more looking at body size, body mass, and behavioral traits, which is a more proximate way of dealing with the challenges posed by changing environment, certainly climate change. So keep that in mind. In Puerto Rico, this genus is represented by 18 species. In total, we have 26 amphibian species, 18 of which are in the genus Eleutherodactylus. We call them in Puerto Rico coqui, which is basically just repeating the two notes of the most common species. 15 are endemic, 3 are listed, one of which is presumably extinct, is this lower jasperite here on your screen, which is the golden looking dude. And then the rest of them are at risk. That means that they could be listed as threatened or endangered as information accumulates. So this is what we call pre-listing conservation to the extent possible. Both the Commonwealth or state agency, Puerto Rico Department of Natural Resources, and the U.S. Fish and Wildlife Service would like to prevent at all costs that we have more species listed as either endangered or threatened. The reasons for this status is that they have all patchy distribution, in part because of habitat degradation and change, but also because they have very narrow range of conditions, especially those 9 species that are high elevation species in terms of relative humidity and temperature. The other one, as I was saying early on, they have high philopatry and poor vigility. So they have two options really. So they stay and adapt, or go locally extinct, or they move, which is what we call positive traction. And movement that is tracking the good suitable habitat. But this option is not readily available for this species. So we're really looking at limited at a translocation as one of our options to try to avert the the status worst status of this species. Excuse me, I went a little blank there. So here's a dissection of what we've done over the last 15 to 14 years, depending on when you want to start. When my clock started in 2011, when we went down to Puerto Rico to consult stakeholders. Out of that meeting, we decided to start with the climate modeling. Puerto Rico was in sore need of fresh and very more reliable climate downscaling data. And that's how we started. So we have multiple people from agriculture all the way to natural resources conservation. The part with the Eleutherodactylus are the coquis, including, as I said, selecting representative species. We did some work with translocations, genetics, and demography. At the end, uh, Mitch is going to cover all this up and give us an illustration of how to inform decisions for decision makers that reduce or manage the risk when you're trying to make a decision, in this case, translocations. As I said, we consulted with stakeholders and we followed pretty much the scheme of this structured decision making. So the first thing we did is we decomposed the problem, try to identify that primary or fundamental objective, and that's what you've got here. And that is to provide actions. And I cannot even read it because everything is blocking, and I want to make sure I read it right. Uh, there you go. Wait, thanks, sorry. There we go. To provide actionable information to conserve and recover endangered and at-risk species, amphibian species in Puerto Rico. We are now dwelling in the second category, consequences, where we're building predictive models, dealing with uncertainty and risk. And Mitch has already started on the third component, which identify prefer action, taking in consideration or being able to articulate what are the trade-offs, constraints, and the benefits. Another way of presenting the result of this consultation is that we have two actions that we want to entertain that are appropriate for the coquis in Puerto Rico. One is managed translocations, and the other one is creation or identification of areas that are always suitable or they're resilient, climate resilient habitat. That's what we call climate refuges. The two questions that will trigger or not decisions by managers is when should I apply or when should I implement some of these strategies and where should I do these? And of course, each one of them has a lot of components to it. And but I'll just briefly go over the top one. So we have information on timing of climate shifts, but that is moderated, if you will, by knowledge on ecophysiological limits. So we want some information on the status of the source population if we're going to do translocation, because in essence, what we're doing is a harvest from a source population to colonize new areas. And then, of course, we need to know something about demography and habitat quality. We follow and we do what we call mechanistic studies, identified underlying factors um, that affect this phenomenon of abundance and distribution, very basic definition of ecology. And then the number two is to test, we also test some predictions about site suitability and validate suitability of these potential climate resilient habitats. So that's sort of our approach. Let's start with what we spearheaded, how we spearheaded this project in Puerto Rico, which was the climate modeling part of it. And we used both a new dynamically downscale, and I'll let Adam explain that if there's somebody interested in the difference between the statistical downscale versus dynamically downscaling data. And then we also use long-term weather station data to get an understanding of where we stand in the recent past. And those are the two major take-home lessons from the climate modeling. Is the current conditions from data using data from 1950 to 2020 indicates that temperatures have been increasing, but mostly at nighttime. And you can see the bottom two lines of this graph. You can see the tendency both at the maximum uh, uh, what is it called? High elevation and low elevation. We have, and the tendency is going up. And this is a little bit on the bad side of these minimum temperatures, because these guys, the Eleutherodactylus, the coquis, are nocturnal species. So this is not necessarily good news if the trend continues over time. The other one on a larger scale was this downscaling model that Adam and Jared Bowden and others put together. And this one is predicting that at least we will have an increase in future conditions of 1.5 degrees. But not only that, we will also have some drying conditions. And it could be substantial, as you can see from this graph. You're going from almost all the way to, I think that probably the average is somewhere between 25, 20% less rain in some of these areas. And this includes the southern, here in the bottom, the southern part of Puerto Rico, which always been dry because of the rain shadow effect. But the other model is suggesting that perhaps we may have some drying conditions that includes the northern part of Puerto Rico, the coast, and the karst belt, if you're familiar with geography in Puerto Rico, and the mountainous region, which is the central part of the island. And I don't know if my cursor, there you go, is showing. One of the consequences could be, for instance, up in the mountains, that cloud base height may be increasing and therefore moving everybody up, if you will, in elevation as they're tracking higher humidity and lower temperatures, and of course, lower precipitation, which affects wetland hydrology. That's a couple of examples. We hope to bring all of this together, the biology, the ecology, and the physics and the climate modeling into maps where we can identify these always suitable habitats. We have here an attempt that was put together by a colleague of mine, spearheaded by a colleague of mine, Marconi Campos Arquera in 2021, along with us, where we identify these habitats in Puerto Rico based on the available knowledge at that point in time. I'm happy to say, if you will, that I listened to a talk from him the other day, and he says, but basically this general location of these areas have survived additional data. So even though he's polished that map, we're still basically looking at the same general areas as being always suitable. So these are areas that conservation planning can look at. More so because we overlaid the protected areas, and about 80% of the habitat here is not protected. So this is something to consider when we're looking at long-term protection and conservation planning, that perhaps we should do strategic decision making into deciding which one of these areas we want to force some level of protection. You don't always have to buy land in order to protect it or provide some level of protection. Let me go into the particular subtopics, and at every point I'll try to relate that back to put it in a larger context, especially with the climate modeling. So thermal limits and sublethal. Here the idea, and I'm taking you back to physiology. Here the idea is to look at what's happened to these animals when they are exposed to these higher temperatures. In one case, we've also done experiment by modifying relative humidity, but this one is focusing on temperature in the interest of time. We are interested in two parameters. One is what we call the high critical temperature. At this point in time, animals essentially stop being able to escape, say, predation or climb up a tree to reach and start calling and advertising. That's because they enter into muscle spasm. It's almost like a seizure. And you know, humans suffer from seizures when temperatures is too elevated, when the high fever, if you will. So it's the same thing here with these animals. So that's sort of an upper limit. But the other one that we were interested in is in number two. It's called Page's range. And Page's is a Latin word that is basically saying things are degrading, they're deteriorating. So the ability of organisms to deal with after being up in the peak, which is the optimal performance, then starts going downhill. And we want to avoid that because although if we keep going, obviously we'll hit that high critical temperature. So keep in mind this is what we're talking about. These two parameters. We use these uh, oven or I call it oven, but it's really heat chamber to determine CT max. We also have respirometers. Here are the frogs inside the tube, the experimental tubes. The one metric that we measure because it was more reliable was CO2 as opposed to O2, but either one can be used for these kinds of experiments. Here are the results of the CT max. Well, what you see here is that by and large, these CT max levels are high. They're in the, and I put it in Fahrenheit so that you can relate that a little bit easier if those of you are not used to thinking in terms of centigrade. But we're talking about 38 for widely distributed species like the common coqui. Here's that red-eye coqui. This is a low elevation, less than 300. Prefers a high elevation, but it's fairly high, 39, 38 degrees. Here are some specialists, and you have here the freshwater wetland obligate, which is the coqui Llanero, and I'll talk a little bit more about this species later on. It's a strict freshwater wetlands, and that's on the coastal areas of Puerto Rico. And then you have the melodious coqui, which is probably the lowest, along with other species that are like this species. Is representative of another 8 high elevation specialists, and their CT max is around 36 degrees. To put it in context, Adam helped us and put a map way back when, or this graph, which I think is very informative. And what he tries to do is try to translate these model outputs into decision relevant information. And what he did here was looking at the proportion of area, 500 meters or above. So we're looking at high elevation species, in this case, the melodious coqui. And he's asking, what amount of that area, what proportion of that area will be exposed to a number of hours over 36, which is the limit for the Eleutherodactylus, the melodious coqui. I want to go scientific. So what we find here, and that's probably good news, is that based compared to a baseline level, which is present or recent in the 80s and early 2000, to the projection 2040, 2060, we have about half the area up above 500 meters will be on the low end of this spectrum. So probably close to zero, maybe 1 extra hour of exposure, which taken as a whole is not bad news for these species, which has the lowest CT max. So this is one good way of putting these things in context. The other way, which is what I was telling you, is that we can measure these sublethal performance with a 24-hour exposure experiments. And that's very important. Again, we have here on the y-axis, we have the excretion of CO2. Here on the x-axis, we have temperature, and our treatments were 15, 20, 25, 30. And you can see these growths are described by, they're exponential in nature, exponential response of the CO2. It increases with temperature. However, excuse me, there are species that have a breakpoint. This is what we call the Arrhenius breakpoint temperature. And what it's trying to tell us here is that internally, these species are juggling on how to deal with additional heat stress, in this case caused by temperature. And so internally, they're channeling or making adjustments to deal with that extra stress that can no longer be dealt naturally by just increasing CO2. And that may have implications, demographic implications, because they may be pulling energy that would otherwise be used, say, for growth, for reproduction, maybe even go out up a blade of grass to advertise for reproduction. So that is very important, and keep that in mind. I'll come back to that in a few slides. So this is the last section of my presentation. I'm going to touch on a few examples on demography, translocations, and genetics. And all of this you'll see suitable for any species, but we're going to focus on a species of the coqui Llanero. So I'll talk about that in a second. So here's a generalized model where you have all the four species of coqui plotted against elevation. I could have used temperature, I could have used relative humidity, but I'm going to touch on that on the next slide. What you have here is that a very nice relationship of going on elevation. So this sigmoid curve here is very representative of high elevation specialists that don't really start picking up in terms of occupancy. Let me define occupancy. Occupancy is the probability of detecting an individual given that they are available to be detected. So it's the probability, if you will, that I have 80% probability of detecting a Watmanai melodious coqui at 600 meters and so on and so forth. So this is a high elevation, very strong sigmoid. When you see an S, that is beautiful. And these kinds of models. You see the opposite, almost a sigmoid relationship with one that loves lower elevation, higher temperatures, and that's the red-eye coqui. And then you have these guys that are widely distributed, such as the common coqui, that are doing fine even at low temperature, at low elevation, higher temperatures, but they love to be up there if they can make it, if they can be there. So this is sort of again relationship of where they are relative to environmental conditions. And here's an important one. Here's I'm talking detecting a chorus. I'll say briefly that we use acoustic recorders for this. So in the spectrogram, you can, if you have very few frogs, up to three individual, you can see the notes because they're almost calling concurrently or responding to each other. So it can be kind of a messy endeavor to try to figure out how many individuals are actually there. But then there can be so many that it saturates the screen and you can no longer turn. And we decided to have, lots of them and not very many of them as two categories here. So we cut that at four individuals, and that's a chorus. And then the probability of detecting an active nest. So we're tying again abundance and reproduction. And what we found, at least in these slopes that were south facing, and we have two of them, is that of course they will be detected in higher numbers or in chorus and reproducing when temperatures are less than 26 degrees centigrade and relative humidity is high around 85. However, you may actually have places or locations within these gradients where you have lots of frogs or cores of individuals, but no activity in reproduction. These are things that we need to sort of consider that you may have one and not necessarily the conditions to have what's important is the fitness component and which is reproduction. So keep in mind, we did this before and after Hurricane Maria, but that's another talk. Lastly, here's the last frog I'm going to be talking about is the coqui Llanero. It has become our poster child. This is for several reasons. It's listed, critically endangered, very isolated populations. And you can see, if you can see this map, you have three populations. They're about equidistant, about 30, 35 kilometers apart. They occur along what used to be northern freshwater wetlands in Puerto Rico. They have been developed, they have been drained for sugarcane and other things historically. Now they have reverted and urban sprawl and other kinds of threats are affecting these wetlands. It's an endemic species, very small. You can see here, the coqui is head to head with FDR there on the dime. So it's a really small dude. Only three populations, as I said. But where we've been able to work with them, they seem to be locally abundant. And that's a really good thing when we talk about genetics. The one thing that we're looking at is, before I do that, I think it was Mitch that put this. I hope you can hear this. Mitch, you're going to be my guinea pig. Let me know if you can hear this. This is the call of the Llanero. Can you hear it? No? Okay, so never mind. Pie in the sky. So
It seemed like it
anyway,
stopped. Maybe hit it again. I mean,
okay,
one more
I'll try
time.
to hit it again and see. Okay.
No, sorry, it didn't
No,
work.
yeah,
Oh, there
okay,
we
you guys
go.
missed it. No, never mind. Anyway, so we wanted to do with this coqui Llanero pretty much what we've done with others: estimate their abundance, identify factors influencing population growth, physiological performance, genetic diversity, because we're thinking about translocation, so we want to know what we're doing here. And lastly, that first point is relevant to the last one, which can we harvest the population? Can we take individuals without undermining their ability to rebound and grow back again and continue, their local persistence? So what we did and discover is that abundance is very much, at least in one of the population, the westernmost population, is, let me just go back here quickly for your benefit. Here's what we're talking about. Here's the story, the work in Arecibo, the westernmost population. What we found is that microclimatic conditions are well within their range of tolerance. What's affecting their abundance is vegetation. It's our habitat component. Remember, I talked about habitat degradation. These places have been, affected or modified for sugarcane plantation historically. Now they're reverting back to vegetation, but it's a mix. It's a patchwork of different kinds of vegetations, and some of the ones that they use. And here, the important ones influencing positively the abundance is things like sedges or scirpus, acrosticum, which is a fern, and then but they don't like, I was going to say hate, but certainly don't like mimosa, which is a shrub. It's very indicative of disturbed areas. So where you have lots of mimosa, you won't find these species. So that we even tested this as preferred species by them. I think what's going on here is not only are they good to go up and down because they do a lot of vertical migration, more so than horizontal migration. Remember, these are very limited in their dispersal ability. Is that the plants are concave, they're axils. So when you reach down, there's a lot of concavity, if you will. So this species lays the eggs in those areas as opposed to all of these vines or mimosa in this case, this shrub. They don't provide that nesting habitat they need in order to lay eggs. The other one was that we were able to estimate, after adjusting for detection probability, the abundance of this species throughout the year. And what we see here is what we expected, some seasonal changes. What was surprising was that the seasonal, we were hoping to get the upper level abundance towards the tail end of the year because it's the rainy season, it's the cooler season, or we're transitioning to the winter in Puerto Rico, which, believe it or not, for tropical species makes a difference. What we found in 2024 was that we had an unusual year. So compared to what we observed early on in the year, which was really unusually rainy, we have sort of a suppression, if you will, or low growth on the population because we had higher than usual temperatures and lower than usual relative humidity. So Arecibo is a unique place, so I don't want to go out of a limb too far. I'll just go out and say that perhaps this is sort of a good glimpse at what may happen with protracted drought periods in Puerto Rico or increasing temperatures and reduction of moisture. And that we may have suppressed reproduction on some of these species, which is usually what happens. So, a take-home lesson there. I'm going back again to that sublethal performance with coqui Llanero, and this is a species that exhibited that ABT or Arrhenius breakpoint temperature. And what's interesting is that we measure this is empirical data. This is not downscale data. This is from HOLS, a little instrument that we use to measure both relative humidity and temperature. And this is nighttime. These two lower lines are nighttime, and they are up and down. This and down would be close to the water in the swamp, which is about half a meter above it, and then the other one is about a meter and a half or so above it. So it's sort of following the profile of movement up and down that these frogs pursue. And what we found is that months like August, perhaps July, are flirting with the idea of 25 degrees. It doesn't mean that they're going to disappear. It just means that they may have to be making some adjustments as they go into these months, and perhaps reproduction, for instance, may be suppressed because they're dealing with these thermal stresses. So food for thought. More interestingly, and if we can put this in terms of projections, Adam again did his wonders in trying to look at what is the average change in evening exposure. And here he's just looking at from 6 a.m., 6 p.m., 18 hours to about 11 p.m., 23 hours in the evenings. And he's looking across elevation. What would we expect in terms of average change in exposure to different temperatures? And he built this graph going from the lowest to the highest treatment temperature that we have in our experiments. And we have, you can see there, increases in addition, we would have about an extra hour of 25 degrees at lower elevations in Puerto Rico. So if you add that to current temperatures, perhaps we may be complicating the fact we're trying to keep this in mind as we're trying to select areas, suitable areas for translocations of these animals. So this is when, again, the physics and the ecology comes together. I'll wrap up with this slide. We've done translocations as the dress rehearsal work for serious translocations, as in the case of the coqui Llanero. We tried it with transmitters. The problem with the transmitter, even though they're tiny, 0.15 grams, is that they're still too heavy for these animals. Here's the melodious coqui down there, and that's much smaller than the common coqui, which can be as big, females can be as big as almost 1.5 inches. So what we were able to track these animals, we were able to get high survival. So what we're doing in terms of protocols is very effective. We can release them in new places, and we can release them in occupied habitats and compare how they move. And results were as expected. Movement will be longer in areas that they are unfamiliar with. The melodious, we went out there without marking these individuals because we haven't found an effective way to have markers that stay with them long enough to serve or for a mark recite or mark recapture approach. But anyway, we were able to track these animals, and that was the beauty of it, that even though they were not marked and it was unoccupied habitat, we were able to track them. The beauty of it is that they survived that initial colonization period, and then they went on and they bred. So we have really a success story in terms of translocation of this species up in higher elevations. Hopefully we can do the same with the coqui. In terms of genetics, we also had good news. We've analyzed 2 populations. We're in the process of analyzing the third as we speak. And the take-home lesson of preliminary results is that high genetic diversity in each population, the mitochondrial DNA suggesting haplotypes is suggesting that they may have been connected. And this is this graph here that looks like a praying mantis or something. And the size of the circles it's sort of the haplotype, the number of haplotypes from different individuals there. It suggested that probably they were connected. These 2 populations, they were probably connected, but we don't know how far in the past rather. So we're looking at probably 100, 200 years. It's pure speculation. We think and we hope that the third analysis of the third population will help us try to untangle this a little bit better. And then lastly, we also look at nuclear DNA, and this one is showing, that's the bar that you see down there. Everybody in the samples stayed within the samples. There's no crossover of genetic material between individuals of the Sabana Seca to the Arecibo and vice versa. And what it's showing is that currently, at least based on nuclear DNA, there's no connection between these 2 populations. Again, the take-home lesson is a high genetic diversity, and we can sample from both, and we could probably bring these 2 species together if we wanted to do a mixed population release somewhere, but that's a decision to be made. In terms of decision, this is where I transition to Mitch, and he's going to give us an illustration of how to do this by assessing risk and informing decision makers. So Mitch, I'm going to stop sharing my presentation, and you're in charge now.
Okay. There we go. Is that slide up still?
Yep, I can see it.
Oh, great. Well, thanks, Jaime. That was a really nice, comprehensive overview of the work that's gone on for the past, what, 10, 15 years, something like that. This has been a
15
really
years, yeah.
15 years. Yeah. So yeah, uh, from laboratory genetics and ecophysiology to field work and trying to assess protocols for doing this management work. It's been a very interesting road, I guess. So, thank you all for attending this, and I'm going to just wrap the talk up with sort of the next steps and what we're thinking about in terms of moving from the science to action. So, as we transition from research to implementation, as Jaime pointed out early in the presentation, the question of where and when to conduct these management actions, specifically translocation, is really the key. We we must consider more than oops, this is not advancing. There we go. We must consider more than just the suitability of habitats or current conditions, because investing in new populations is hopefully a forever prospect, or at least a very long-term one. So that question of the future becomes really important. These considerations must account for where future conditions are likely to become favorable in the future, also where favorable sites may become less so, and the velocity of climate or other changes such that we can predict or estimate when is best to act. So the problem, of course, is this unknowable future. We don't know how fast, how much, or where these conditions will actually change. And so this huge amount of uncertainty in the future, of future climate change in particular, represents a great deal of risk when we're talking about very costly decisions of investing in protected lands or the protection of lands, the translocation to establish new populations, etc. If we could somehow manage these risks, we would have a lot more confidence in selecting these sites and moving forward with using this best science to try to protect these endangered species. So fortunately, and this will be just a quick overview of kind of the theory behind this, we are able to borrow this theory or theory from other disciplines to manage this risk under climate uncertainty. And this is kind of the key. Economics, in particular, has provided us with tools to formalize the diversification of investments such that we can reduce the risk of uncertainty in financial markets, right? That's what economics is working on. And the most important of these theories is something called modern portfolio theory, which was developed by Harry Markowitz in the 1950s. So, to make the connection, any good financial advisor, thinking about your retirement accounts or whatever, will urge you to not put all of your investments, all of your eggs, for your retirement funds in Apple or some other historically high yielding stock, but instead to hedge your bets by directing some of your portfolio to potentially lower yielding assets such as, municipal bonds. What your manager is really asking you to do is to accept lower returns to help safeguard against market volatility, which we know is a real important thing, such that when stocks fall, typically bonds do better and vice versa. So we translate the ideas behind this investment portfolio diversification to the spatial diversification of potential conservation lands that we might consider for restoration, for translocation, or other actions that might be needed to establish new populations or manage existing ones. So in spatial planning, we look for parcels to invest in that are negatively correlated or at least uncorrelated, much like stocks and bonds, such that under one future climate scenario, a portion of those parcels will be better for these frogs than others. And under a second possible future, maybe the tables are reversed. But the goal is that whatever future unfolds, some of those parcels in our conservation portfolio will always be good for frogs and therefore contribute to the species persistence. So for any possible portfolio that of parcels or other assets that we would consider, we need to measure two things to do this in a formal, logical way. The first is the benefits of the portfolio, which is really the composite value of all the parcels in that portfolio, averaged across the uncertainty of possible future conditions. And this is what we consider or call the expected value of the return or of the benefits. Um, on the other hand, risk is quantified by calculating the variance of these individual assets as, under these different climate scenarios, as well as the full correlation structure across the portfolio that accounts for both those positively and negatively correlated assets or parcels in terms of what they might do in the future. So these two metrics, the total return and the risk, are in conflict, right? We usually, and thinking back to economics, higher returns usually mean greater risk, and some lower risk portfolio will often result in lower benefits, right? We trade these off. And so to address these trade-offs and identify a portfolio that might be optimal that balances these two objectives, risk and benefits, we plot using those two metrics that I mentioned, all the hypothetical portfolios that we could put together, that we could imagine. And we plot them against these two axes, and then we take advantage of something called Pareto theory to identify potentially optimal solutions. So this efficiency frontier is defined by the portfolios that can't be improved upon without either giving up some level of benefit or accepting some additional risk. So all these solutions that are found along this frontier, which is actually created by the potential portfolios as I defined it, they are considered to be optimal. And so for these, trade-offs are really based on the values or how much risk the decision maker is willing to accept. So all the portfolios that we might consider below this frontier are considered to be inefficient or dominated alternatives. These are defined by portfolios that can result in a better return that can be found for the same level of risk, or conversely, a portfolio that has less risk for the same amount of return, right? That we don't move up in that expected return axis. Okay, so the lesson on portfolio theory is over. So along with the other components of the work that Jaime just described, we began to develop a decision model framework and we used, as Jaime had mentioned, this poster child of coqui and Llanero to build this framework. And this was built around the express management need to establish new populations for the species that only currently exist in three small populations, as Jaime mentioned. So to identify the suitability of future refugia or habitats or parcels, we modeled the relationship between a number of environmental conditions under multiple future scenarios. All these things Jaime touched upon, and so this is where those data come into this model, and we estimated the future Llanero distribution under these conditions. The conditions and our predictions were generalized at this point, generalized over a uniform grid of coastal parcels below 100 meters above sea level, which is where we find the coqui and Llanero. And the conditions were predicted, or the benefits were quantified, I should say, as the cumulative occurrence probability estimated for each cell in a given portfolio of parcels, as well as a function of a range of possible actions that could be applied to that cell. And so we input these data into our relatively new spatial R package that allows us to run this portfolio evaluation for all possible site combinations, like wherever we could potentially do a translocation or a restoration of habitat. Here's just a single example of some of our modeling of future conditions under uncertainty. These were relevant to changes in amphibian distribution along the coast, specifically for this coqui and Llanero. These figures demonstrate the predicted changes in wetland extent, as well as the change in Llanero distribution near the three currently occupied sites that Jaime pointed out earlier. These are done under three different scenarios of future sea level rise. Uh, because we won't know future emissions, the extent of this habitat reduction or change is completely unknown at this point. And so the idea here is that we need to capture this uncertainty for those predictions and consider all of these, the potential outcomes of these conditions as crucial information for selecting these long-term translocation sites where we might put these frogs now and hope that they persist into the future. So the actions and the cost of those actions were considered as a function of the parcel classification, right? This is a very important component of the cost and the potential benefit of selecting a particular parcel, whether a cell is currently protected or unprotected, and also whether it is an existing wetland or one that has been degraded due to those impacts that Jaime mentioned, agricultural development, hydrologic manipulation along the coast. The status of the parcel determines whether the parcel might be added to the portfolio or ignored, and which other actions and associated costs might be available, and hopefully to determine which ones would be optimal for each parcel selected in the portfolio. So these actions that we consider include a combination of whether we invest in protecting a parcel that we might use, whether we maintain an existing parcel, whether it's protected or not, whether we conduct a restoration, which would be a costly action, whether we do the translocation, and in addition, how much we expend in monitoring costs and effort. So the size of a conservation portfolio, of course, as you might imagine, will also depend on the available budget that the managers have. And so at this point, we don't know what those future budgets might look like. So we also have to model that kind of uncertainty. So we started by analyzing a simple cost-benefit trade-off, considering risk at sort of a moderate level, or the risk attitude. So we know here that as budgets increase, the optimal portfolios of selected parcels move beyond those currently occupied wetlands on the north coast and begin to increase in diversity further from these known areas of historical occurrence or distribution. And we note that as budgets increase, optimal portfolios move beyond those currently occupied wetlands and begin to diversify further from these known areas of occurrence. We then selected a single budget scenario, in this case, a moderate budget figure, and explored the effects of that values-based risk attitude on the optimal conservation portfolio. So represented here from top to bottom is showing increasingly higher risk aversion by decision makers. So thinking about having less and less risk as we move from top to bottom. So we see briefly just a significantly greater spread of selected parcels in the portfolio as that risk aversion increases. And we assume that this is a function of the distance and an assumption that as distance increases between parcel pairs, they become more negatively correlated, right? And so this is our hedging bets as risk aversion increases, and to make sure that if conditions are not what we hope for in the future, that there'll be some parcels that are actually doing better than others, right? That's the bet hedging. So finally, to add increasing realism, we include the full range of land classifications, whether they're protected or not, as well as those available actions and an estimate of the relative cost of those actions. So this is an example of a single optimal portfolio solution with a relatively large budget, and selecting a sort of a balanced midpoint in terms of risk preference along that Pareto frontier. With the decisions, of course, being to act on or ignore each possible portfolio or possible parcel in the portfolio. So we can see here that there seems to be a shift in investment focus towards the eastern portion of the north coast, with those blue and green cells indicating recommended translocation sites, as well as potentially investing in new habitat conservation. So putting habitat areas into conservation, and this, possibly reflecting some sort of east-west gradient of future habitat, climate, and coastal change conditions. We also see that there's some recommendations to forego restoration of drained wetlands, and even to abandon some sites that are currently occupied in the far western population of Arecibo. That's on the left side of the map here. Although there are a few sites along the west and central coast that remain apparently viable for some conservation attention. So we note here that this, importantly, that this decision model is very preliminary. We're using a number of assumptions and not necessarily all of the data that we have at hand, as well as some potential all different objectives of the managers that we could then add in here. But the idea here is that this represents a decision framework that can incorporate, say, new covariate relationships. As we develop them, we can include, the actual costs of a parcel to put into protection, as well as the cost of these various actions. And again, as I mentioned, other possible objectives that we obtain from the Department of Natural Resources or the Fish and Wildlife Service and other decision makers as we learn more and move closer to actual implementation. I'm going to hand this back over. I can just leave this slide up, but I'm going to hand this over to both Jaime and John, who is Sagara, who I believe is in the audience now, representing the Fish and Wildlife Service in Puerto Rico, to talk a little bit about next steps and answer questions. And I think Adam will join this as well. So Jaime, do you want to just sort of walk through the next steps for us?
Yeah, I'll do that. Glad to do that. I want to, since this is being taped, I want to correct something, clarify something. I mentioned earlier, and I think I got confused by the slide, reading the slide. The first attempts or the oldest fossils of colonization by Eleutherodactylus actually date back to 29 to 47 million years ago. That's a long time ago, but it's certainly not 200 million years ago, as I first said. So that's I got a little bit confused there with the number of species in the area as opposed to the millions of years that they've been around in the Caribbean. So excuse it, I just want to set the record straight. Okay, yeah, so we're reaching a point where we can start putting together some strategies, and as Rich is saying, inform some decision makers as to what to do. So what we're doing as a first step, and here I am joined by, being led by my colleague Nathan Hostetter, who is also with the co-op unit, Mary Diaz, who was a student that worked with the Llanero over the last 2 years, and along with other colleagues, including John Segarra from Puerto Rico. Hopefully, he's one of the participants, and the rest of the team. And that's that we've reached the point where we probably ready to take action. And I don't think we will have time for Q&A. I'm sorry. But anyway, we'll take action, and we're assembling a team just like we did earlier with this project, where we consult everybody. And then, and before we take any actions, everybody's in agreement. We reach some consensus. We are starting to seek funding. We got partial funding to initiate at least one first experimental release. They are planned to start in 2027. 50 individuals, those are details, and then monitoring the short term. And then last but not least, I don't know what the bullet point was there, the last one, but basically, we hope to continue doing work along the lines of what we're doing in helping Puerto Rico Fish and Wildlife Service meet their managerial needs and conservation needs. So thank you very much for listening. I'll regret that it took so long, that it went over time, I guess.